In part 2 of chapter 6, we'll talk about the adaptive immune system and then some ways that viruses can evade immune responses. So just to remind you, in part 1, we discussed the innate immune system, that immediate, non-specific reaction to viral pathogens that in turn activates the adaptive immune system, which is stronger response, specific response, and potentially a much longer lasting response. And as you can see here, the adaptive immune system involves primarily T cells and B cells. So let's look at T cells first. So there's really two main types of T cells, two different classes, and they both use what are called T cell receptors to recognize viral antigens. But the two classes are the cytotoxic T cells and the helper T cells. And the cytotoxic are so named because they can induce apoptosis in infected cells, seen here, and they utilize this helper molecule called CD8 that will help facilitate binding to antigen by the T cell receptor. So these are sometimes called CD8 T cells or cytotoxic. And then the helper T cells, their main role is going to be to activate antibody-producing cells called the B cells. And they also have T cell receptors that bind antigen, but their helper protein is called CD4. So these are CD4 T cells or helper T cells. And we'll look at how both of these work here in just a second. So if you remember the dendritic cells from the innate immune system, those were the phagocytic antigen-presenting cells that would ingest an antigen and then present them using what's called the major histocompatibility complex, the structure designed for presenting antigen. And there's two different versions of the major histocompatibility complex. There's class 1 MHC or class 2 MHC. Class 1 is recognized by CD8, so dendritic cells presenting antigen using class 1 MHC will activate the CD8 cytotoxic T cells, whereas dendritic cells presenting antigen with MHC class 2 will activate CD4 helper T cells. But you can see here what the activation would look like, right? You've got the dendritic cell with that major histocompatibility complex presenting the antigen. The antigen directly binds to the receptor on the surface of the T cell with the assistance of either CD4 or CD8, depending on which T cell type we're talking about. And so these interactions are very specific, right? So each T cell receptor is basically designed just to recognize one specific antigen. And you can imagine, well, there are lots of different possible antigens out there. So pretty amazingly, during embryonic development, your body produces just a staggering number of different T cell receptors through a gene rearrangement process. And Dr. Peak in the biology department is actually something of an expert on that phenomenon. So if that's something you're interested in, you could pick her brain on that. But basically, your body is capable of producing a billion different T cells, right? So that is a lot. Each one just capable of binding an individual specific antigen. So we have this antigen specificity. And binding of that T cell receptor to its antigen then causes that T cell to activate and proliferate, make copies of itself. So it would look something like this. So imagine you've got a dendritic cell presenting this antigen using MHC class 1, right? It's no match for the first T cell, no match for the second T cell, but on the third, we get a match, we get binding, you can see the T cell is activated, it's going to proliferate rapidly, make millions of copies of itself, that all have the same T cell receptor that would then be capable of identifying that same antigen. And those T cells, some of those are going to return to the infected tissue. So remember, this kind of matching process is likely happening in the lymph node. So those new T cells that are produced, in this case cytotoxic, uh, leave the lymph node, go to the infected tissue, and they are going to look for cells in that tissue that are presenting the same antigen using MHC class 1. So basically all nucleated cells in your body can produce that major histocompatibility complex, class 1, and if they are infected with a virus, they will present viral antigens on their cell surface. And so you just have this army of T cells looking for, okay, the dendritic cell told me it found this viral antigen. Let's go find cells that are showing that same antigen. And when they find them, they are going to kill the cell, right? These are the cytotoxic T cells, thereby hopefully eliminating some or all of that viral threat. So just go back to this same idea of cell killing. You remember one of the innate cells was the natural killer cell. And what it's doing is it's actually surveying tissues and inducing apoptosis in any cells that it sees that do not express the major histocompatibility class 1. So if we go back to the cartoon here, we should see really all cells should be producing MHC class 1. Whether they're infected or not, or presenting antigen or not, is another story, but all cells should have MHC class 1 on their surface. And if they don't, that may be an indication that they're infected, because a lot of viruses have evolved such that they can basically inhibit MHC class 1 production, right? That would be a solid strategy if you're a virus. And this was kind of the cell's response is, oh, okay, well, if that's the case, we're going to send in these natural killer cells that are going to basically destroy any cell that doesn't have MHC class 1. So I'm going to show you what it looks like, how a natural killer cell decides. It's actually sort of a two-step process, because you have receptors on the natural killer cells that are both activating and inhibitory. I'll just show you the image. I think that'll make more sense. So here's your natural killer cell. It's got an activating receptor and an inhibitory receptor. And so if it sees a cell that has an activating ligand but also expresses MHC class 1, it binds, but that inhibitory receptor prevents the natural killer cell from killing. Whereas if it binds to a cell that expresses the activating ligand but does not have MHC class 1, that would be suspect as being infected with a virus, so that will trigger apoptosis through the natural killer cell. So again, that's less specific, right? It's part of that innate system, but it's easier to talk about here after we've already introduced this idea of MHCs. Okay, so we had the cytotoxic T cells being activated as part of this adaptive response, and then we have the helper T cells. And as I mentioned before, the job of the helper T cells is basically to activate antibody-producing cells, which are the B cells. So there's really only a few cell types that express MHC class 2, which remember is what's used to activate the helper T cells. MHC class 1, as we just discussed, should be found basically everywhere. So you're really only going to find MHC class 2 on B cells, dendritic cells, and that's really about it. Some macrophages as well. So the B cells, the antibody-producing cells, have to be activated by a helper T cell first. And what B cells are doing, B cells can identify antigens, even whole viruses, so they have B cell receptors on their surface that could bind to, for example, an entire virus. They can internalize those antigens and then express using MHC class 2, either part or all of that antigen. So in this case, right, binds the virus, internalizes, breaks down, and then loads at least a portion of that virus or viral protein into that MHC class 2. Um, and it is now going to present that to helper T cells. Okay? And if we have a match, if we have some binding, then we'll get activation of that B cell. And it would look something like this. Okay, um, so we'll go back, remember we have our dendritic cell, innate cell, presents an antigen to the CD4 helper T cell, activates it, right? We had a match between the receptor on that helper T cell and that MHC class 2 antigen. So it becomes activated. And in the meantime, we had the B cells out here that had also found that same virus, right, and are presenting the same antigen that the dendritic cells found on its MHC class 2. So that helper T cell that's activated, it now goes looking for B cells that might potentially have that same antigen. If it finds one, it binds, and now the B cell becomes activated, and it will start producing copious amounts of antibody, which can bind to and hopefully inactivate virus particles. Okay, so once we have these B cells that are activated and producing antibody, they're considered plasma cells, and sometimes you'll see antibody production referred to as the humoral response. But we have this sort of confirmation, right? The dendritic cell finds an antigen, activates the T cell. T cell is then shown the same antigen by a B cell. So now the B cell is active and we're really off to the races into the thick of that adaptive response. So in terms of antibody production, there's a few different flavors, classes of antibody. They're technically called isotypes that can be produced. The first kind that gets made is what's called the immunoglobulin M, IgM. It's always the very first made. If you detect IgM antibodies, it's indicative of a new early stage infection. Over time, we make slight changes. We tweak the antibody to be more specific. Also, sort of trim it down so it becomes a little bit smaller, and we get these higher affinity immunoglobulin G (IgG) antibodies. These are smaller. They can cross the placenta and go from mother to child and provide some immune protection there. There are a couple of other isotypes. There's IgA, which gets made at mucosal surfaces, can be in breast milk. IgE, which is associated specifically with allergy response. All right, so what are those antibodies doing? Well, as I said, they could bind specifically to the viral antigens, right, viral particles, and just by nature of being bound, they can inhibit the antibody or the virus's ability to bind receptors on the cell surface. However, if you remember macrophages, some of those phagocytic innate immune system cells, they have antibody receptors on their surface. So if, in going about their business, if they bump into an antibody, they can bind and then ingest whatever the antibody is bound to, so taking it out of the picture. And likewise, the natural killer cells that we talked about, again, innate immune response cells, they also have antibody receptors. And so in this example, you have a virally infected cell that is producing some viral antigens, viral proteins on the cell surface because it's getting ready to bud off new viral particles. So the antibody that was being made recognizes that antigen, binds to that infected cell. Now the natural killer cell can bind to that antibody, and that will activate apoptosis and destruction of that infected cell. Now, we were just talking about dendritic cells, so they're activating, just to kind of summarize this whole process one more time. Dendritic cells see that antigen, they can activate the cytotoxic T cells in the lymph node, proliferate, go to the site of infection, and then kill via apoptosis infected cells that are presenting same viral antigen on that MHC class 1. Those dendritic cells can also activate helper T cells with MHC class 2. Um, those helper T cells can in turn activate B cells, which begin producing antibodies, become plasma cells. Um, Antibody progresses from IgM to IgG, and those plasma cells can crank out antibody for weeks or months. Once the infection is cleared, then your body doesn't have a need for this huge army of plasma cells, or even activated lymphocytes for that matter. So a lot of those T and B cells are going to undergo apoptosis. They're just not needed anymore. However, some small subpopulation will remain alive. They will become what are called memory cells. So um, those memory cells are the basis for immunological memory, right? And that's really the power of the adaptive immune system is if you see the same pathogen months or years after the initial infection, you will get a secondary response now that is not only much faster, right? You can see we're jumping into the adaptive response very quickly, but the magnitude is much higher. And that's just because you've got larger numbers of memory T cells and B cells already present, compared to the uninfected, naive, quote unquote, naive cells. Those T cells and B cells that are present don't need as much activation, so we get antibody production happening more quickly. And we can already make the higher affinity IgG antibodies rather than the IgMs. So yeah, big advantage to having that immunological memory. And of course, this is the whole basis of vaccination, right? You're intentionally introducing virus or virus antigen in a harmless form so that you can get that stronger response when the actual pathogen shows up, if it shows up at a later time. And depending on the particular virus, and the number of times that you've been vaccinated, that immunological memory could last for the rest of your life. In some cases it won't, right? You have to go back after a certain number of years to get a booster shot. It really just depends on the particular virus. Okay, so to finish this chapter out, let's talk about some ways that viruses can evade these immune responses that we've been covering. So we already talked about the idea that it's a sort of an evolutionary arms race. But one way viruses can get around immune responses is by directly encoding gene products in their genome that will produce proteins that can directly interfere with immune pathways. And there's all kinds of examples of that, specific proteins made by viruses that block various aspects of the immune response in some way or another. A second way they could do it is through what's called antigenic variations. These would be small mutations that are basically leading to the production of new antigens that are no longer recognized by the antibodies that are being produced by those memory cells. So this would be also sometimes called antigenic drift. So in this example, right, you've got the virus on the left here, and your body is producing antibodies that recognize the proteins on the surface of this virus. Well, now we have a mutation in the genome of this virus that has slightly changed the shape, the character of those viral surface proteins, and has changed them enough so that they still work for the virus's purposes, but the antibodies that your body's producing that are very specific no longer recognize it. So from your immune system's perspective, it's might as well be a whole new virus. So that's a good way for viruses to get around it. We already talked about the fact that especially RNA viruses mutate very quickly. So that's very common. Third way is what's called reassortment. This really only applies to viruses that have segmented genomes, but this is even more dramatic than antigenic variation. Instead of an antigenic drift, it's called an antigenic shift. So we have a larger change. And in this case, this requires a cell to be co-infected with, for example, two different subtypes of a virus. And you can see this virus has a segmented genome. right? Multiple nucleic acid molecules. So if you have both of these subtypes in one cell, carrying out their replication cycles at the same time, when this infected cell goes to package up new virions, um, it needs to have the correct number of genome segments, but it's not going to be capable of distinguishing segments from one subtype or the other. So it's just going to randomly grab, the number that it needs, um, and that may include molecules from both subtypes. So now what you end up with budding out of this infected cell is something, a virus that doesn't, at least in terms of its antigens, does not resemble either subtype 1 or subtype 2. So this would be a new subtype 3, right? And most certainly your immune system is not going to be able to recognize that new subtype. So this happens with influenza virus all the time. And then the last one I wanted to mention is what's called latency. So this only applies to certain viruses that are capable of going dormant, like herpes virus, go through these periods of latency and then reactivation. But because when these viruses are latent, they're not actively replicating, there's just no antigens present, right? The virus is basically hiding out, and there's just nothing there for the immune system to detect. So these are some of the arrows in the quiver of the viral defense response. All right, thanks for hanging with me. I know the immune system is complicated, but hopefully you got a decent overview there.